And our next speaker will be Joan Slonezewski, with the zoo in it. Joan Slonezewski. She's a professor of biology at Kenyon College, and her talk is titled Designing Humans for Beyond Earth.
Thank you very much for inviting me to this meeting. It's the first visit to a Space Society meeting. I'm a microbiologist. I do molecular biology on E. coli, the bug that took out a town in Canada. So, I'm not sure what I'm doing here, except that one advantage of going to space is I hear there's no E. coli out there. So, of course, we all bring about 10 to 14 cells of it along with us, but never mind. Okay, so my topic today is about life shaping, what I call the genetic engineering of people to live on other planets. But before we get to that, though, I would like to make one last comment about the previous presentation. If you're interested in stem cells for a newborn baby, for children born today, there's a very easy source of stem cells, which is the umbilical cord blood. And if you have a child born today, you can store the stem cells forever from that child. So it's something for you to think about if you want to save for potential replacement organs. That's something that, as parents, you can do as of now. Very exciting technology. Now, why would we want to think about genetically engineering people for other planets? Well, it's interesting to me as a science fiction writer as well as a biologist that in science fiction, certain problems are never really addressed. When you see people visiting other planets, unfortunately, the Star Trek model is kind of the accepted one. In most science fiction stories, they'll work out all the orbits very carefully with all kinds of calculations of the sort that Robert Forward does. And then they'll have people step right onto a planet where there's green grass, and they can eat the food there, and even mate with the inhabitants, and so on. What is wrong with this picture? Well, there are a number of things. I would say that when people talk about the problems of life in space or life on other planets, there are three categories of problem. The physical, the chemical, and the biological or biochemical. Now the physical, I think people get that pretty straight. I mean, there are programs on the web you can do to calculate, what kind of planet could be habitable for humanity from a physical standpoint, having a certain range of gravity, a certain spectral range of the sun, a certain size of the planet, orbits, and so on. That's pretty well worked out. Then you get to chemistry, and people have a generally pretty good idea that you need to have some oxygen around. Okay? But when you get to biochemistry, there's silence. People don't think about the biochemical issues. So what I'd like to do is first talk about the biochemical issues, and then and show why we need to think about genetic engineering, and then see what kind of genetic engineering we might do to address those biochemical issues. Okay. We all know that life is made up of certain essential biological molecules, and for simplicity, I'm going to refer in particular to the components of DNA and the components of proteins. We all know, thanks to Jurassic Park, about Mr. DNA. Okay, and so here are some examples of, here we see the adenine, cytosine, guanine, and thymine, the basic building blocks of DNA in life on Earth. Is there any kind of a pointer? Sorry. Okay, so now the interesting question is, if you went to another planet, would that other planet use the same basic building blocks of life? Okay, we're assuming now that you go to a planet that if it's habitable, presumably, I'm assuming that living things will have evolved to live there already. If life hasn't evolved, probably it's because it can't evolve. Okay, so if you go to a planet, there are living things. If we go to Mars and find Martian microbes there, will they be like us? Well, one theory says that yes, they will be like us. They will probably use the same bases of DNA that we use. And if we look at the amino acids, they'll probably use the same amino acids that we use. And this theory goes that life evolves in the best possible way, the sort of Voltairean view that life has always evolved in the best possible way. There's an optimal way, that there's an optimal information storage molecule, and that would be DNA, and the molecules that form proteins are the optimal ones, so that any living system in the universe will use the same 4 building blocks of DNA and the same 20 amino acids. That's one extreme of the sort of universal view. And there's some evidence in support of that. Okay. For example, if you ask what happens in early Earth, so-called early Earth experiments. Who's heard of early Earth experiments? You know, where you boil up, we've all heard this, boil up certain molecules. Well, it's really freaky that when you do this, if you boil up molecules for a month under reducing conditions that you would expect in early Earth, what forms spontaneously? Adenine. I mean, you look at what a complicated molecule that is, the basis of ATP, the basis of DNA, and it forms very straightforwardly from 5 molecules of hydrogen cyanide. You can see, if you count the atoms, it just takes 5 molecules of hydrogen cyanide spontaneously to form that molecule. Furthermore, if you look at meteorites that come to Earth, they show traces of adenine in it. So this is powerful evidence that adenine is likely to be something that would have formed spontaneously, and whatever living cells evolve, they're likely to use adenine. Similarly, if you look at what amino acids arise spontaneously under early Earth simulation experiments, there are 5 amino acids that arise, glutamine, proline, and so on. And those same 5 amino acids are the same ones that are found in meteorites, and they're the same ones that form the base of the biochemical pathways. So it's like there's a message here, okay? That adenine is basic. There are these amino acids that are fundamental. So, we can be optimistic and say that wherever we go, in the universe, we're going to find the same building blocks of life. We can eat the food and so on. Okay, so that's the optimistic view. Now, the pessimistic view, however, is, let's look a little closer at this. First of all, that's only one DNA base. There are 4 that we use, but in nature you find other bases in minority amounts that could just as well have been used, only slightly different versions. If we look at amino acids, some amino acids are essential. That simply means that these are ones that our bodies have forgotten how to make, so we have to be sure that the plants we eat have them. The so-called non-essential ones are the ones we can make ourselves. If we look at these so-called essential amino acids, we see some pretty complicated molecules. Furthermore, if we look at plants, which we eat, we find that plants make about 100 different kinds of amino acids, some of which are actually toxic to us. And there are all sorts of variants on amino acids. That's why you have to watch, being a naturist, what you eat from nature, because there are things very closely related to our amino acids that are, in fact, very toxic to us. An example of that is, in particular, the amino acid tryptophan, which is one I'm very interested in my research. The amino acid tryptophan looks exactly like alkaloid poisons. It's very closely related to alkaloids and things that cause hallucinations like LSD, neurotoxins. So if you just rearrange a couple of atoms on the molecule, you get a poison. However, suppose you had a biochemistry evolving on another planet where just by chance it evolved to use some of those poisons as its basic molecular structures, and then the other amino acids either wouldn't be present or they would be toxic. Okay, so there's no absolute reason why an ecosystem evolving somewhere else would use exactly the same amino acids we do. In fact, it's far more likely that there would be slight differences on the molecules here or there that would result in a new ecosystem being totally toxic to us. It would be full of things like the amanita poison from the mushroom or snake venom. You know, what poisons are is molecules that look a lot like biochemicals, only just slightly different. That's what makes them a poison. So we're going to be going to a planet that's full of poisons. Okay, so this is the sort of pessimistic view, and this is the view that I address in some of my novels, which I've listed up there, in case you're interested in stories dealing with these issues. So this view would say that we go to a new ecosystem, it'll be totally different, or different enough to cause us problems. We won't be able to eat the crops that grow there, and so on. Okay. On that planet, though, you know, all the organisms would have evolved to be able to eat each other. It wouldn't be a problem there, but it would be a problem for us. Okay, so what would we do about that? Okay, there are two general ideas in science fiction for what to do about this. And the most commonly proposed idea is that of terraforming the planet, meaning, okay, if we can't live on this planet, we're going to just boil it up completely get rid of the biology and the biochemistry and replace it with our own ecosystem. Okay? Well, that's one thing. One way to go. How difficult might that be? Whether it's ethically defensible, those are other questions. So the alternative might be, could we possibly genetically engineer what I call life-shaping in my novels? Could we life-shape the human colonists so that they could survive on this alien ecosystem? Well, suppose we try to do this. How might we do this? Okay, first of all, okay, one goal could be if we could add genes into our genome that would encode enzymes to detoxify the components that were toxic to us. That would be the most obvious thing. Now that might take a lot of genetics, maybe not, okay? If we think about where we're at now, we're starting to talk about genetically engineering humans now. I'll get to that in a little bit. This is not unfeasible. Even with present-day technology, this is not unfeasible. Second, more interesting, could we add genes to people's genomes that would actually use the alien biochemicals to convert them into things that we need to eat? Now, you remember that we noted that there are essential amino acids and non-essential amino acids. The reason some are non-essential for us is that our bodies have forgotten how to make them during evolution. But we could re-engineer our bodies by putting in genes for enzymes that would create those amino acids that we need. Now, what would it take to do this? Okay. Well, until very recently, we would have said this was hogwash. But in the past 5 years, the Genome Project has led to incredibly exciting possibilities. Not only can we see the entire genome, but the cloning technology has enabled us to manipulate it. One of the interesting things we found is that the genome has surprisingly few genes in it. It's amazing how few genes it takes to make us up. They used to think 100,000, now they're down to 40,000. You know, as they're sequencing more and more of the genome, they find that there are these vast stretches of unused DNA separating a handful of genes. I mean, 40,000, you know, how many people are there in Tucson? You know, 40,000 is a small number. So it may be that this is a manipulable project. Now, how do we go about manipulating genes? We have various kinds of technology using pieces of DNA called carrier DNA or vectors. And some very exciting vectors are coming out. Some of you may have heard of plasmids. Who's worked with plasmids? I bet some of the high school kids have worked with plasmids, and you're snipping the DNA with restriction enzymes and ligating it. Okay. Well, there are new generation vectors that are being devised which enable us to insert genes into people's bodies. grown people. You don't have to wait for the next generation. For example, some of these vectors are based on AIDS virus. The viral cause of AIDS is particularly good at manipulating human genetics, and we can tame the AIDS virus and convert it into a carrier for genes that we want to carry them into people's body cells. Okay, so the gene manipulation technology, this is already being used to cure blood diseases, to treat various genetic deficiencies. So that technology, gene therapy, is here already. So the technology is here. Now, how are we going to do it? Where are we going to get the source of genes for enzymes that are going to help us survive on other planets? Okay. Well, here's where it starts to get a little bit dicey. And the question is, how far do you want to go here? Whose genes do you want to have? Okay. There are various ideas of where to get genes to help improve human function. The first idea, of course, is, let's find humans vary, of course, humans vary tremendously, and different ethnic groups happen to have in their genome different genes that are useful. For example, there's a family in Italy that has a gene that enables them to drink red wine and eat red meat and never get heart disease. It's called the sin gene. And so anybody that has this gene, there's this one protein it encodes, okay? So wouldn't you like to get genetically engineered with that one gene, okay? And that's something that medical companies are working on. Now there are ethnic groups throughout the planet that have these particular genes that just by chance in nature possess the capability to protect us from problems on our own Earth. So some of these might incidentally protect us from poisons we find on other planets, okay? Now, you wouldn't mind getting a gene from some other ethnic group if it was going to help you live longer, right? Or survive on another planet, right? You wouldn't mind that, right? Okay. Now, another source of genes to help us improve our function is our nearest relatives. The chimpanzees, it turns out, are 98% identical to us genetically, and they actually have a superior gene pool. It turns out that humans underwent some sort of an evolutionary bottleneck about 100,000 years ago, and we ended up like the cheetahs. We're basically clonal compared to other organisms. I know that's hard to believe because the differences that we see appear so magnified to us, but all the ethnic and racial differences are less than you would expect to find in a population of other organisms. And as a result of going through this bottleneck, we have lost a lot of genes that we ought to have to protect us from various things. For instance, cancer. Chimpanzees are much less susceptible to cancer than humans are. They have all kinds of genes that protect them. So there's research done saying, well, let's take chimp genes. If we provide people with chimp genes, they can be protected from cancer. Anyone care to volunteer for that? Would you take a chimp gene if you could prevent cancer? Okay? Okay, we're still happy with that. Okay, so we don't just have to look at other ethnic groups. There are our nearest relatives in the animal kingdom. They have genes that are worth it. If we go a little farther than chimpanzees, we can look at, for instance, the New World monkeys. The monkeys of South America are a little farther related than us. They have the prehensile tails and so on. But they also have digestive systems that can digest all kinds of toxins found in plants. Plants make chemical defenses. And these digestive systems are among the most robust of the primate world. And so if we're going to visit other planets, we probably ought to get ourselves engineered to have howler monkey digestive tracts, so that that could be the way to help us better digest what we find out on this other ecosystem. Okay, any volunteers for that? Okay, oh, well, this is an adventuresome group. It's worth the prehensile tail, isn't it? Actually, the prehensile tail might be even more useful in weightlessness, as a matter of fact. Okay. Well, I get the idea that's what this conference is about. It turns out, in fact, that any gene in the entire gene pool, we're starting to think of the entire gene pool as the entire gene pool of the entire biosphere as the joint resource, the joint inheritance of all creatures on Earth. Because it turns out that genes can be taken from completely differently related organisms and can be made to function in a new organism. So we could, for example, if, talking about telomeres, no sweat. Well, you just heard a session about telomeres. If we want better telomeres, why stick with primates? Why stick with animals? Why don't go straight to the sequoias and use their telomeres? Okay, DNA is DNA. If telomeres are really the answer, which, by the way, I'm not quite sure about that, but that's another lecture. However, if telomeres are the answer, all we have to do is snip out a few telomeres from the sequoias, and you wouldn't mind sprouting a leaf now and then, would you? Okay. So these are some perhaps wildly speculative, but perhaps not so speculative possibilities. Now, one thing though is it does take time. Genetically engineering humans takes time because of our long reproductive systems and various protocol problems. It might be faster, instead of just engineering us, to engineer bacteria that live within us. All of us possess 10 times as many bacterial cells in our bodies as human cells. They live with us symbiotically. And symbiotic bacteria can do all kinds of things that we can't do. For example, supposing we wanted to engineer ourselves to do something totally radically different, such as breathe a different molecule than oxygen. Suppose we wanted to breathe methane and eat sulfur. Okay? Now, this gets outside the realm of what animals and plants can do, and would probably be hard to engineer ourselves to do. But bacteria are a different story. Okay? If we look at bacteria, bacteria have all kinds of ways of getting a living. And I'm reminded of Tom Janquish's lecture about the rockets. Rockets are basically involve a fuel that has to burn. Well, our digestive systems are basically the same way. We have to eat a food, which is a reducing agent, and oxidize it with this complicated electron transport chain in between. Okay, now most of us eat sugar and breathe oxygen. However, bacteria have different ways of doing it. Okay? For example, there are bacteria that instead of using sugar as a reducing agent, they can oxidize hydrogen sulfide or methane or even molecular hydrogen, hydrogenotrophs, methylotrophs. Okay? And all it takes is just one or two enzymes at the beginning of the electron transport chain to make that possible. Furthermore, some of these bacteria, where you can find them symbiotically with organisms, with fairly complex organisms in the deep sea thermal vent ecosystems. You can find crabs that look just like sea level crabs, giant clams that look like sea level giant clams. But they have symbiotic bacteria in them that use hydrogen sulfide and oxidize it with oxygen. So they have both hydrogen sulfide and oxygen in their bloodstream. And that took just a little bit of evolution, not even very much. So perhaps if we could engineer bacteria to eat the hydrogen sulfide, we could eat hydrogen sulfide on this new planet instead of bothering with the local flora and fauna. Going even further, there are bacteria that don't have to breathe oxygen. They can breathe sulfur, using sulfur as their oxidant, or they can even use metals such as uranium or gold. Now this is not science fiction. There are bacteria that get along by oxidizing using ions of uranium or ions of gold. In fact, there's a hypothesis now that all of the gold metal that exists is the waste product of microbial bad breath. So if you don't want to breathe oxygen on this new planet, maybe this is where you need to go. And I think I'm going to stop here and see if there are any questions. I think that's enough speculating for one day. 100,000 years ago? Well, that's right. The evolution, basically every organism that evolves as the result of countless cycles of growth exponentially followed by near extinction, because evolutionary change can only happen in a small population. And in the case of humans, it happened apparently in a particularly small population based on mitochondrial evidence, mitochondrial DNA evidence, and so on. And so that would have been about 100,000 years ago, maybe maybe a couple 100,000, it's hard to say. But the fossil evidence bears that out also. So that there was a relatively small population. It could have been as much as one family. Okay, there could have been, it could have been, the Eve hypothesis. You could have had one family that had major changes in the genome that led to it being reproductively isolated from other apes. And then the various enhancements of brain capacity and so on could have happened by just tiny bits of fine-tuning of the genes, the ape genes that were there. Yes?
Millions of years to make humans and all the other species, don't they much more likely to add a gene, a single gene that would kill someone rather than make somebody happy? I mean, like levels of just sugar. I mean, if you added a gene that made sugar out of something else as well, and you increase the amount of sugar, and diabetes is caused by just
Right.
problems like
You're saying this is a kind of a dangerous proposal, and that's a very good point. What you're saying is exactly what the NIH study committee will say when anybody proposes this experiment. These are very tricky things to do, but it all depends on how badly you want to do it. If there's a compelling enough reason to do something, people want to do it. In terms of saving lives, these gene replacing things are happening right now, because if a life is at stake, you're desperate to do it. So the technology of gene therapy that's going on right now, in terms of actually manipulating people, but that's also a reason why I like the bacterial idea, because manipulating the bacteria and getting them to survive in experimental apes first would be a pretty good bet that they would help humans survive also. Yes?
When you talk about inserting genes into the human, do you insert it into the existing human in many different locations, or do you insert it into the reproductive cell in the
Okay, so you're talking about two different possibilities. You're talking about somatic cell gene therapy versus germline gene therapy. And that's an interesting question. Right now, well, first of all, both are possible. Okay, right now the greatest interest is in somatic cell gene therapy because, for one thing, you can target the particular tissues that you're interested in. In my books, I deal with both. I deal with somatic as well as germline gene therapy. I mean, the question is, do you want to go to... One of the things the points I didn't have time to get into was, you go to this new planet, you can live there, but can you bear children there? Okay? When the Spaniards first came to Central America and tried to colonize the high plateaus of Peru and so on, they could live there, but they had to send the women down to the bottom of the mountain for their confinement because they were unable to give birth at the top of the mountain, whereas the natives who had lived there for many generations had evolved to be able to give birth in the high altitude. Okay, so we're going to face a similar problem when we go into space. We may live there, but will we be able to maintain pregnancies to term and raise children in those environments? And that will be 100 times harder than anything that Shannon Lucide goes through to live in outer space. Yes.
You mentioned 10 times as many bacterial cells as human cells.
That's correct.
Which side of that is mitochondria?
Okay, our bodies contain 10 times as many independent bacterial cells as human cells, most of them living in the gut. The bacterial cells are smaller than human cells. That's kind of how that works out. But I'm not counting mitochondria. Mitochondria are unavoidable symbionts of the cells. Yes. how many of those gut cells do we depend on for metabolism? Well, it's not very well understood, but for example, we depend on our bacteria to make vitamin K. That is why in hospitals now, in our infinite wisdom, we emphasize sterile birth. I mean, there's a reason why babies are born in the place they are, because they have to get colonized by bacteria to help them out. But of course, we have sterile birth in the hospitals, and therefore we have to inject babies with vitamin K, since they don't yet have the microbes to make it, okay? So it's very, very interesting ecologically. Vitamin K for sure we need from bacteria. There are other vitamins that we probably also get as well. It's no problem. The bacteria will join you in space, I guarantee it. That will not be a problem. I think we need to move on.
Yeah. Thank you, gentlemen.